want to start by asking you to think about how you got here tonight. Not in some philosophical way. Another speaker would be a better person to help you with that. I want you to think about how you actually travelled here to the Royal Institution this evening. If you're like me, you might well have used Google Maps or City Mapper to help you find your way. Being a civil servant, I probably have to say other mapping applications are available. And if you're joining us online, welcome to you all. You might also have been using a device like your phone. You're probably watching on a phone or a tablet or on a laptop. Either way, most of us will have got here with a bit of help from a device like this. And I want you to think a little bit about the phone, the battery in your phone. Chances are that it's a lithium-ion battery. And if you came here in an electric car or on an electric bus, that also would have been powered by a lithium-ion battery. Now, lithium-ion batteries have many advantages. They hold lots of energy and they're easy to recharge. Those of us old enough to have used the old-fashioned alkaline batteries, the kind that you put in the back of a TV remote still, will remember those terrible moments when your device ran out of power and you had to go scrabbling around the house to find enough batteries to put back into your device so that it was working again. Our phones may go dead, but it's simple and convenient to recharge them. But there are downsides, and particularly there are downsides in all of the metals that go into making up these lithium-ion batteries. So that includes lithium, cobalt, and other rare earth elements. Getting hold of these metals is hard. Most are currently extracted and then purified from rocks in the ground, and those are processes which are very energy intensive and very polluting. And recycling and reusing these same metals is also hard. This is the periodic table of the elements. So it's created by Dmitry Mendeleev, published in 1869, first presented in London here at the Royal Institution 20 years later. How many elements do you think are used in electronic products? Well, it's often 60. Electronic products can contain up to 60 different elements, around 52 of them being metals. Those are the ones highlighted in blue now. And we currently rely on inefficient and environmentally damaging methods to isolate and also to recycle those individual metals. Indeed, many electronic items can't be recycled, and they simply go to landfill. So this is already a serious issue, and as global demand for electronics grows, it will be a growing problem. Well, what if I told you that researchers here in the UK have identified naturally occurring bacteria which have the ability to extract and recycle metals from this sort of waste? Hats off to anyone in the audience who's heard of the bacteria Shigella oneidensis MR1. It is capable of extracting manganese from lithium-ion batteries, or of Disulfovibrio alaskensis, which is able to precipitate cobalt out of mixtures of metals and other chemicals that can be found in these kinds of batteries. Now, I'm only aware of these bacteria thanks to amazing research taking place in the UK, including by Louise Horsfall's group at the University of Edinburgh. Louise's team have been collaborating with researchers from across the country as part of the ReLib project, which stands for the reuse and recycling of lithium-ion batteries. Actually, one of the funders of this project is the Faraday Institution. That's the UK's flagship funding programme for battery research. And of course, it's a great delight to be talking about that right here at Michael Faraday's desk. So on his desk, somewhere, I do have a few items to help us talk about the process that Louise's team use. They are primarily focused on recycling the metals that are found in the enormous batteries that you would use to power a car or a bus. That would be too big for me to bring here. And actually what I have, if I could have the visualizer on, please. What I have, how am I doing? This is so difficult, this bit. Is this is some of, there we go. This is some of the a recovered recycled lithium-ion battery. So that's actually part of what's called the cathode of a battery, which is very rich in the metals that we're interested in. So once lithium-ion batteries reach the end of their life, what you do is you take them apart, you dismantle them mechanically, and then you shred the material, and you'll end up with something a bit like that. What you do next is, either using biological or chemical processes, you dissolve the metals that were in the battery in order to have them in solution, so make what this stuff here, which is called metal leachate. I'm not managing very well, am I? There we go. So that's metal leachate from, what have I got to do? I've got to go that way. There we go. So this bluish liquid is metal leachate from a lithium-ion battery. And it's to that liquid there that you would add bacteria that are able to take up the metals that are now in solution in there and then secrete them as individual metals, so in tiny nanoparticles of metal. And from that, you could then recover, one by one, the metals that you want, including, for example, manganese here. I told you earlier that the first bacterium I named was able to remove manganese, and that is manganese removed in Louise's lab using the processes I was just talking about. And eventually, when you've been through that process a couple of times, what you end up with is this. This time it's a slightly straw-coloured liquid, which is called a lithium biobrine. So this is very rich in lithium. It's actually rather similar to what you would get from a lithium mine, from lithium deposits in a South American lithium mine. And that, too, can be used to make new batteries. So with a process like that, you can, in fact, create a circular economy where instead of throwing away your battery, you can create a new one from it. And I'm not just talking about a few types of microorganisms to improve the extraction and recycling of one or two metals. There appear to be lots of different microbes out there capable of extracting different metals. Indeed, it's possible that these bacteria have evolved this capability in a way that detoxifies their own environment, collecting up and excreting harmful metals so that they're not poisoned. So if we use combinations of these bacteria and we tweak the characteristics of the strains that we can find in nature, we can increase the efficiency with which metals are purified and recycled from waste. And that word tweaking is important. And it doesn't really do justice to the science involved, because what we're really talking about is engineering existing microbes to extract and recycle metals. Extracting metals from the ground is hugely expensive and a damaging process. It looks rather like this. What you see in the bottom half of this picture is an open-cast manganese mine in Ukraine. And once we finish with these products, needing such metals, we throw them away. The top part of this slide shows a landfill site in Ghana after a fire. And there have been reports of lithium-ion batteries causing fires in landfill sites right across the world. With engineering biology, we only need to remove metals from the ground once. Thereafter, they can become part of a genuine circular economy through continual reuse. So we can use physics, chemistry, engineering to get them out of the ground in the first place, but then we can and should use biology and engineering to keep recycling them. And this is just one example of what is within our grasp thanks to the power of the science called engineering biology. I'm speaking about engineering biology this evening because I believe it could be the most significant branch of science for decades to come. I want to explain why I think that's the case and to share my excitement about this field for two main reasons. So first of all, the science is absolutely beautiful. And secondly, perhaps more importantly, is that both the current and the future applications of this science will make a huge difference to the everyday lives of people in the UK and across the world. I'm here to convince you of both of these things, but if I can only win with one of them, it will be the second one. I would like you to go home thinking about the usefulness of engineering biology. I'm really keen for people to recognize that the scientists and engineers working in this field are working to produce solutions that most, if not all of us, would view as necessary, some of them urgently necessary. So let me kick off by explaining a little bit about what I do. I'm Government Chief Scientific Advisor, so my role is to advise the Prime Minister and the government on any matters related to science, technology and engineering. The job and the advice is a mixture of proactive and reactive work. It'll cover everything from providing scientific and technical advice during a national emergency to explaining the risks and opportunities around emerging technologies like artificial intelligence and engineering biology. So now, in getting to grips with engineering biology, I had a small head start. I am myself a mathematical biologist, so sort of halfway there. And my own research focused on using mathematical models to understand the evolution and the spread of infectious diseases like measles and like HIV. I don't, however, have any background in engineering, nor in biochemistry. So I have had a steep learning curve to climb to understand these technologies. So at this point, let me explain what I have learned that engineering biology actually is. Engineering biology involves applying the principles of engineering to the manipulation of biological processes in order to bend biology to our will. In other words, it's the practice of using ideas and tools taken from engineering to design and modify living organisms or biological systems. Using tools and ideas developed over recent decades, the goal is to develop new materials and energy, to improve animal, plant, and human health, to address environmental issues in new and sustainable ways. So what we're talking about is the ability to harness and control biology predictably, repeatably, and, as I've already said, usefully. Sometimes that will mean working with what's already available in nature, and at other times it will involve genetic modification techniques. So let me unpack some of that a bit further. Firstly, on the engineering side. So here I want to start with the design, build, test, learn cycle, sometimes called DBTL. This approach has been central to product design and engineering across many engineering disciplines for some time, and it drives continuous refinement and innovation, making research and development faster and more efficient. In engineering biology, design, build, test, learn is brought to bear on biological processes, by which I mean any activities occurring within living organisms. Essentially, I'm talking about designing something biological, like a version of a cell, or it could be a biological process, such as cell division, or a genetically engineered system, then building it, probably in a laboratory, and then testing it to see how well it works, before finally, and perhaps most importantly, learning from what didn't work, and then feeding the lessons into another round of design, making improvements again and again around this cycle towards a well-defined end goal. This looks like being a more efficient way of recycling metals, to use the case study I gave at the start. And why is this approach necessary? Well, because living organisms are highly complex, with many different parts and networks of interactions between those parts. One could argue that physical or chemical systems are a bit more straightforward, more predictable, more easily quantifiable. We've been using this design, build, test, learn process to bend chemistry and physics to our will for more than a century, very successfully. The complex and less predictable nature of biological systems means we need to work through multiple permutations to get to a desired outcome, and that's where the engineering, in engineering biology, comes to the fore. If we can get this approach right, and I'm going to offer further examples where we are getting it right, then we have the power to systematically develop biological systems to meet some of the biggest challenges we face. Let me be more clear about what I'm claiming. If the 19th century was the golden age for chemistry, and the 20th century the same thing for physics, then this century ought to be the golden age for biology. What right do I have to be so optimistic? Well, I'm going to go through some of these truly astonishing advances that have been made that make me think that these things are within our grasp. So where should I begin? Well, of course, we have spent thousands of years modifying the living world. And I'm not going to go all the way back to our first attempts to domesticate wild crops. I'm not even going to go back to Darwin and Mendel. And instead, I'm going to start with Watson, Crick, and Wilkins. And I'm glad to say I do also have a picture of Rosalind Franklin on there. I was delighted to hear her name checked during your demo just now. 3 of these 4 received a Nobel Prize in 1962 for determining the structure and code of DNA. And what they did, we can think of as discovering the language of biology. Understanding the structure of DNA opened the door to reading this language, then editing it, and then actually starting to write it ourselves. Our ability to read DNA took many leaps forward, thanks particularly to the work of Walter Gilbert and Fred Sanger. They shared half of the 1980 Nobel Prize in Chemistry. They had done a lot of work to understand the building blocks of DNA, the nucleotide alphabet of biology, if you like. During the early part of my career, the standard processes for reading gene sequences were called Sanger sequencing. Our ability, well, the next game changer after that was in 1983, when an American biochemist called Kerry Mullis invented a process called the polymerase chain reaction. It's better known as PCR, and it's a lab technique that's used to make copies of very particular pieces of DNA, and you could think of it as a photocopier, a very specific photocopier for DNA. And it's really important because it lets scientists easily and cheaply create many millions of copies of a particular stretch of DNA, even when the original amounts there are only very small, and then those can be read. So it makes reading the DNA in a sample possible, even if it's only there in tiny amounts. You'll all have become familiar with the PCR during the COVID pandemic, because that was the test that was early on used to make many copies of the viral genetic material in a throat swab to allow reliable diagnosis of a COVID infection. That was the test where you did a swab, popped it in a test tube, and then stuck it in the post, and hopefully got an answer a few days later. It was particularly important early on, before we had home testing kits. The invention of PCR also earned a share of the 1993 Nobel Prize in Chemistry, so that's DNA Nobel number 3 on my list. So if we fast forward to 2003 and we get to the completion of the Human Genome Project. So researchers across the world spent some 13 years cataloguing the precise sequence of all the DNA in the cell of a human being. It was a huge effort. I already said it took 13 years, and the sequencing of that first whole human genome cost about £2.5 billion. Thankfully, and also remarkably, sequencing technology has come on leaps and bounds over the past 20 years. Now it's possible to sequence the same amount of DNA as in that project in a single day. So that's down from 13 years to 1 day, and at a cost of a few £100s, so down from £2.5 billion to a few £100. So I have here a portable sequencing machine. This is made by Oxford Nanopore. You simply add your sample in here, just in there, shut it in, turn it on, plug it in directly into your laptop, and it will read the DNA sequence of the sample you have put in. This is particularly useful for situations where we can't send off a sample for analysis and wait days for the results. So if, for example, we urgently were trying to identify the cause of an infection in some far-flung part of the world. So we've learned to amplify DNA using PCR, and we've learned to read DNA fast and cheaply using rapid sequencing technologies. We've also started learning to accurately and precisely edit DNA. Previously, when we started to do this, the methods were somewhat crude. There were these things called gene guns, which actually what they did was they fired, what they fired was gold nanoparticles coated with DNA into tissues. One of the things you'd fire them into, for example, would be a tobacco leaf. And some of that DNA would make it into the nucleus of the cells that you wanted, and somewhat miraculously end up in the right place. That's not what we have to do anymore, because we now have tools like CRISPR-Cas9. So this is another Nobel Prize-winning technology developed by Emmanuel Charpentier and Jennifer Doudna. So what they can do is take a targeted piece of DNA and change it very accurately in specific places. Some people have compared CRISPR to using a pair of genetic scissors. So think what a big step forward that is. You go from shooting DNA into a leaf to snipping DNA using molecular tools inside the nucleus of a cell. Some of you might be wondering what's the difference between engineering biology and another common term, synthetic biology. They are often used interchangeably, and different countries use them in different ways. The way I see it, synthetic biology refers to tools like CRISPR, used to design and build new biological components, whereas engineering biology is taking these tools, with or without genetic modification, and using the design, build, test, learn cycle to apply these tools at scale to find solutions to problems in the world around us. There are still challenges with the accuracy of such tools, but the possibilities are truly vast. We know that certain diseases are caused by mutations in a single gene. Sickle cell disease is a well-known example. It's caused by mutations in the beta-globin gene, resulting in red blood cells which are misshapen. As a result, these red blood cells don't flow around the body like they should, and this can cause those affected, and that's roughly 17,500 people in the UK, to suffer from anemia, as well as complications like terrible pain and organ damage. In the past, the only treatment was to rely on regular blood transfusions or a bone marrow transplant, neither of which comes without risks or complications. However, researchers have been using CRISPR to precisely edit the gene responsible for sickle cell with great success, so much so that in January this year, the treatment was approved for use in the NHS as the world's first gene editing treatment for blood disorders. And this is just one of many gene editing clinical trials going on right now, including treatments for liver disease, heart disease, and some cancers. And the possibilities are not confined to human diseases. We can use these genetic scissors to develop crops that are better at withstanding drought or more resistant to insects, so we don't have to rely so heavily on pesticides. And it's these tools that are being used to modify the bacteria designed for metal recycling that I spoke about at the start. Now, it would be remiss of me to talk about the tools of the future without mentioning AI and the transformative impacts it could have. And a prime example is the challenge of understanding and predicting how proteins fold up intricately and precisely in all living processes. Decoding this process was something that scientists have been trying to achieve for many, many decades. It was probably the biggest outstanding single problem in biology, so famous that it had its own name. It was called the protein folding problem. As I say, had stood for 40 years. And then in 2018, DeepMind came along with its AI model, AlphaFold. AlphaFold has since been used to calculate the structure of hundreds of millions of proteins. And yes, it's earned the UK's Demis Hassabis a share of last year's Nobel Prize in Chemistry. So to recap, in the last 60 to 70 years, we've gone from discovering the structure of DNA to learning how to read gene sequences, to learning how to amplify DNA so we can read them when they're there in very tiny amounts, read the first full human genome, and then massively sped up and made it much cheaper to read a gene sequence. And then more recently, we've learned these tools for very precise gene editing techniques, before just recently using AI to make clear and useful predictions of protein structure after reading gene sequence. All that's missing on that timeline is the capacity to design a new protein from scratch, de novo. That will bring us into the realm of being able to write the language of biology, designing and printing a sequence of synthetic DNA to produce a protein with the properties that we want from scratch. I've just been talking about how technologies like AI and tools like CRISPR have broadened the range of biological tools we can use to increase our ability to design and optimize biological systems. And all this comes with valid concerns about risks. An example, a true life example, which springs to mind, is the story of some scientists in Australia who created a version of a mouse virus, this was back in 2001, that instead of causing the normal, mild symptoms, caused all the mice to die within 9 days. Actually, they were conducting some completely innocuous genetic engineering research to try and make a mouse contraceptive. It was a mouse contraceptive vaccine for pest control, and inadvertently found a way of creating a much more deadly version of the mousepox virus. Unsurprisingly, when this research was published, it caused a dramatic stir, a lot of discussion about should the work ever have been published. In my opinion, it's a very good thing that it was published and very widely discussed, and I think it's important that we know about what research along these lines is being done. The point I want to make is that we must develop the right practices and regulations so that we ensure that research is carried out safely and responsibly, but we do not stifle all innovation. We refer to this as responsible innovation, and it's one of the pillars of our government vision for engineering biology, incredibly professionally steered by my colleague Izzy Webb, who I'm very glad to see in the audience this evening. The call for responsible innovation has given rise to new guidance on which genetic sequences people should be allowed to order for their research, and that is welcome progress. Having the UK take a lead in this kind of responsible innovation, where we're thinking carefully about the desired benefits of our research, as well as about how to avoid the negative impacts, lets us manage the risks and harness the wealth of opportunities that engineering biology can offer. There are also other challenges. What's standing in the way of exploiting all this engineering biology for good? I'm not going to spend very long on this because really I'd like to speak to you about the amazing scientific possibilities. But it is important to talk about the barriers. So we've already spoken about proper regulation for engineering biology. We also need to have proper ways of funding the basic research that drives this wonderful technology, and also the application of that research that lets us solve real-world problems. Then there's also the task of making more people aware of the potential for progress here. And a key area for me, and a common issue across all areas of science and technology is making sure we have the right skills in our future workforce to perform the future jobs that come with new technologies like these. The skill set for engineering biology is particularly broad. The field is a combination of many different skill sets and mindsets. Mostly we train people either to become biologists or to become engineers. And for this, we need people who can think with both those mindsets. So we need to think about a pipeline which starts in schools, with children still getting the right grounding in the key disciplines, but also children hearing about the exciting careers available through developing and using the technologies I've talked about. I think it's vital that we don't only think about technical skills. Communication skills are also extremely important. It's a wonderful thing to do pioneering, cutting-edge research, but we also need to be able to explain what that's about and why people should want it. So I've told you a bit about what engineering biology is and how we've got to this point, poised for this biological century. And I've also talked a bit about risks and challenges. But now I think it's time for me to turn to delve further into the applications that I think are so inspiring. Today, I launched a report called Engineering Biology Aspirations. It's our attempt in Go Science to share our excitement about the possibilities that this technology opens up. And we want to share it with everyone, my colleagues inside government and also with a much wider audience. It contains case studies written by UK-based experts that illustrate some of the diverse problems we can address using engineering biology. Microbial metal extraction is just one of them. I want to highlight some others during the rest of this hour, and to recognise some of the amazing research taking place here in the UK. One of the reasons that I commissioned the report is that I think all too often, when someone mentions engineering biology or synthetic biology, the examples given will involve vaccines and medicines. Of course, those are fantastic, important applications. With the COVID pandemic just a fresh memory, we are all acutely aware of the life-saving importance of rapid and effective vaccine development. And I am genuinely in awe of those researchers who can edit the gene that causes sickle cell disease. But I do want to make sure that we also shine a light on the true breadth of opportunities that engineering biology presents, not only in health, but also in agriculture, materials, chemicals, energy, and defense. So let's shift gear and think about the fashion industry. Unlike metal recycling, it's a sector familiar to all of us. We all buy and wear clothes, and we don't often stop to think about where they've come from, how they've been made, and at what cost to the environment. Putting aside issues around workforce conditions and waste in manufacture, the fashion industry is one of the world's largest polluters, responsible for up to 8% of carbon emissions globally. Not to mention the pollution generated in the form of clothing and textiles dumped in landfills like this one, which is in Bangladesh, never to biodegrade. And at the same time, 1/5 of the pollution of clean water around the world is caused by dyeing and treating textiles. And then there's also the growing awareness of the environmental damage caused by microfibers shed from our polyester clothing. So it's no surprise that plenty of researchers and companies here in the UK and beyond are seeking inspiration from biological processes to make new materials that don't rely on fossil fuels or on animal products such as leathers. You may have been wondering why there are bottled drinks and a jolly nice handbag here on the Faraday desk. Well, they're made of essentially the same material. The process of making both these items starts with starts with microbes that naturally produce a material through fermentation called nanocellulose. And that's what, you can see it fairly well here, that's what produces the jelly lumps in this very sugary drink. I've got some here to show you, so that if you can't see them, these lumps of jelly. So it's a completely natural product. It's made by fermenting coconut water. And what's in that jelly lump is a biologically derived nanocellulose. It's a company that I visited last year is called Modern Synthesis, and it's based in South London and founded by Jen Keane and Ben Reeve. And they're aiming to develop scalable solutions to meet the fashion industry's need for high-performing, versatile materials that don't pollute the planet. So what they do is they take these nanocellulose fibers, pretty much exactly the same stuff, and then they process it, and they coat it onto fabrics like linens and cottons, and that way they make new composites. Those are then finished with natural coatings like waxes and oils to improve the performance and to enhance look and feel, which are, of course, very critical to customers. And the result is fine handbags like this. Not yet on sale, but watch this space for next year. And on the slide behind me, you can see in more detail the fibers that make up the handbag. These minuscule nanocellulose fibers are actually extremely strong, weight for weight, 8 times stronger than stainless steel. And Modern Synthesis is just one example of a pioneering UK company making waves in this area. Another example is Selena Materials. They're using AI to help design completely new materials from scratch, including fibers that are very effective at absorbing energy. This makes those fibers particularly relevant for the military and the police who need blast, ballistic, and stab-proof clothing. I used to be the chief scientific advisor in the Ministry of Defence, so it's a particular joy for me to see engineering biology provide solutions for problems in the world of defence. Developing new materials like these can significantly reduce greenhouse gas emissions compared to traditional material production. So this includes minimizing the environmental impacts of raising livestock for leather, or the energy-intensive processes involved in creating synthetic textiles, such as polyesters and nylons. And better still, these materials can be designed for complete biodegradability, getting away from the big problem of plastic pollution. So let me quote for a moment from our report. Imagine a world where every piece of your clothing has minimal cost to the environment, with zero waste going to landfills. Even if a piece of clothing is accidentally discarded into the environment, it safely biodegrades to leave no trace of its existence. This is the future of fashion, and engineering biology is helping to make it happen. So let me move now to another pervasive problem, and that's inefficiencies in food production. Most of you will be aware that fertilizers are used by farmers across the world to apply nitrogen to their crops. Without fertilizers, yields suffer, and we don't have enough crops to feed ourselves. But there are 2 problems. First, the process for making nitrogen fertilizers is very energy intensive. It's responsible for between 1% and 2% of the entire world's energy use, and it generates matching carbon dioxide emissions. And secondly, when you come to use these fertilizers, you again have considerable environmental impacts, releasing further greenhouse gas emissions and damaging waterways thanks to fertilizer runoff from fields. This slide shows excessive algae growth. That's not a path, that's a river. That's a river absolutely full of algae because there's been too much fertilizer runoff, particularly nitrogen runoff, from the fields around it. That is, in fact, the River Wonsum in Kent. Currently, farmers use more than 200 million tonnes of chemical fertilizers every year around the world. Now, don't get me wrong, this ability to produce nitrogen at scale via the Haber-Bosch process was without question the most important chemical breakthrough of the 20th century. The reaction that underpins this industrial process is shown behind me, converting nitrogen and hydrogen into ammonia, which is commonly used in fertilizers. It was discovered by Fritz Haber, and over half of the global population depends for survival on foods fertilized using industrial production of nitrogen. But for the reasons I've outlined, we do need to do better. So how can engineering biology help? What if we could engineer cereal crops to absorb their own nitrogen from the environment without relying on fertilizers? We call that fixing nitrogen. And there are actually examples of this happening in nature. There are bacteria in the soil called rhizobia, which are particularly good at fixing nitrogen. In fact, they convert nitrogen gas from the atmosphere into ammonia, which is precisely the form of nitrogen that plants need. Legumes such as peas, clover, and lupins all do this. They attract these rhizobia bacteria to live in their roots in small structures called nodules. So in return for a steady supply of ammonia, the plant houses and feeds the bacteria, forming an ideal symbiotic relationship. Behind me is an illustration of a plant root with nodules on it. But in my own personal homage to Blue Peter, I have actually brought some clover with me. So that is a plug of clover out of my own lawn at home. And if earlier on, took some of those roots out, washed the mud off them, and then put them onto a dinner plate with a bit of water on them, if I could have the visualizer on again, please, you would be able to see clear as day that there are little lumps on it. So that's just off the clover in my garden. You'd have the same thing if you've got clover in your lawn. Maybe you have better lawns than me. So these little lumps here are exactly what I was talking about. So those were made by the plant, but they're full of bacteria that have come to live there in this symbiotic relationship between the clover and the rhizobia. Lost. So for these sorts of plants, we can coat their seeds with bacteria, with rhizobia, and get great increases in yields that way. Or we can put bacteria out straight into the soil in a process that's called soil inoculation. However, the problem with cereal crops like wheat, barley, and maize is that they don't have these root nodules, and they don't have any way to produce the signaling chemicals that legumes use to attract the rhizobia. So here's another plant that I brought from my garden. This does actually give me pain to take this one out. So this is a baby sweet corn plant. And if I take it out of its nice little thing, and you can see its roots there. I don't actually have enough water to wash them off. But again, in true Blue Peter style, I have one I prepared earlier. And this one's harder to see. But there are, not least because it's hard to see nothing, it's probably, we think you get better resolution there, but there are no nodules. Because this kind of plant simply doesn't make that kind of symbiotic relationship with bacteria. So what researchers like, so here's a photograph in case you couldn't see it properly in the real one. So there are root nodules on the clover, and there is a different maize plant. I've sacrificed so many maize plants. A different maize plant with no nodules on it. So obviously, you can see where this is going. What researchers like Phil Poole at the University of Oxford are doing is trying to engineer a new generation of fertilizer-free crops. And they're going to draw on plant genetics, on biochemistry, and soil ecology. And one of their approaches, given what I've just described, is to engineer cereals to form just these kinds of nodules on their roots that can then host nitrogen-fixing bacteria. The UK is leading the way on this. Oxford and Cambridge universities have major programmes backed by investment from our research councils and by the Gates Foundation. And in fact, the teams involved work together as part of a large collaboration and have recently made some significant advances. They've engineered barley to form nodule-like structures, and they are engineering barley roots to release the chemical signal rhizopene, which prompts rhizobia to start fixing nitrogen. That design, build, test, learn cycle that I talked about earlier is an absolutely essential part of this research. All of the progress made so far has built on round after round of modifying, testing, and redesigning organisms. There are still many hurdles to overcome, both from a technical perspective and, of course, societally, because we all know that genetic modification of crops is a very sensitive issue. But the value of the prize here is large, and I think scientists should not be shy about describing it. Because imagine a world where humanity's main source of carbohydrates, cereal crops like wheat, barley, and maize, are able to generate their own nitrogen fertilizer. We could tackle global food shortages on a much more sustainable basis, and at the same time solve one of the most urgent climate challenges, consigning industrially produced nitrogen to the past. Now, let's just think about crops in a further context, because harvesting doesn't have to be the end of their engineering biology journey. At the start of this talk, I name-dropped a couple of bacterial strains in relation to metal recycling, and here comes another one. This time, it's a type of bacteria called Halomonas. Researchers like Nigel Scruton from Manchester, who I know is here and I can't see him because you're all dark for me, are engineering these bacteria to act as efficient factories for converting food waste into fuel via fermentation. When I say factories, I'm not talking about the massive industrial sites we would normally associate with fuel production. This photo is of Fawley Oil Refinery in Hampshire. To get a sense of how huge it is, look at the cars at the front of the picture. That is a massive site. By contrast, these fuel-producing bacteria can be housed in different size containers, like the ones on this slide, some of them not too dissimilar from shipping containers. The beauty of this technology is that it is inherently portable and scalable to meet demand, with transformative implications for remote areas of the world where energy infrastructure can be scarce. And crucially, these are cleaner, fossil-free fuels that can be used to power homes, businesses, even aircraft. Let's focus on that last application for a second. Burning fossil fuels is generally accepted as the main cause of global warming, so it's essential that we find ways to transition to sustainable sources of energy. Engineering biology solutions like Nigel's can therefore play a significant role in creating a future without fossil fuels. So imagine a world where clean fuels could be produced locally and on demand, including in all those remote and sparsely populated regions of the world which currently struggle to access the fuels they require. Now, I argued just a moment ago that I wanted to convince people that engineering biology is about much more than just vaccines and medicines. And I hope that I've surprised at least some of you with the breadth of examples I've described so far. But I do have one example from medicine that is just so fascinating, I can't leave it out. And that's research into laboratory-grown blood. Why would we need such a product? Currently, the world relies entirely on human blood donations to treat disease and for emergency medicine and surgery. In many countries, including the UK, donation rates fluctuate, and that means shortages can happen. On top of that, donated blood has a limited shelf life, and it's challenging to store, and it's challenging to distribute. When you consider the fact that some countries don't have the infrastructure to deliver blood products safely, or think about conflict or humanitarian emergencies, the problems associated with donated blood become even clearer. And there are a few more besides. It can be very difficult to source some rare blood types, and although blood services do, of course, screen to avoid known pathogens, there's always the risk of new pathogens arising and being passed on to patients who receive blood transfusions. For all these reasons, finding new ways to produce blood would be another game changer, and once more, engineering biology can help us. Researchers like Ash Toy at the University of Bristol are exploring the possibility of banking unlimited supplies of red blood cells, either by transforming stem cells or genetically reprogramming donated precursor blood cells. What you see on this screen is a very beautiful illustration by artist Claudia Stocker, which provides a visualization of CRISPR, the genetic scissors technology that I mentioned earlier, being used here to edit the genetic material of a precursor cell that will go on to become red blood cells. The part of the image to focus on is the blue circle in the middle. That's the cell that will eventually give rise to the red blood cells around the edge of the picture. The little blue donuts represent the CRISPR technology in action, actively and precisely editing the DNA exactly as it was instructed to do. This editing can let us produce precursor cells that can grow and divide indefinitely in a controlled environment, giving us unlimited blood supplies. The Bristol team pioneering this research has been working closely with NHS Blood and Transplant and other partners in a groundbreaking clinical trial that's called RESTORE. RESTORE is the acronym for Recovery and Survival of Stem Cell Originated Red Cells. It's the first time in the world that red blood cells grown in a laboratory have been given to another person as part of a trial into blood transfusion. You might have seen media coverage of this program, which has attracted interest from all over the world. The trial should produce its next set of results by the end of this year or early next year, so it's worth keeping your eyes open for those results when they come out. In the future, we could go a step further and use CRISPR to delete the genes responsible for blood types, for blood groups, and in doing that, we could create universal blood that would be invaluable in providing blood transfusions for individuals with rarer blood types. So this slide is a brief reminder of the complexities around ensuring blood compatibility between donors across the top and recipients down the rows. And what you see is that if you have O-negative blood, you're a fantastic blood donor because your blood can be given to anybody, but you're a pretty poor blood recipient because you can only receive O-negative blood. So you can see that's why what we would love to be able to do is in the lab create unlimited amounts of O-negative blood. The prospects here are, again, tantalizing. Imagine a world where no patient dies due to a lack of compatible blood following an accident or during surgery, where safe blood is available on demand, can be stored for longer, and is free of disease transmission risk. So, there are all these amazing opportunities, which you can tell I greatly enjoy talking about. We've covered a fair bit of ground about engineering biology, not just historically, but geographically, in universities and in companies, and across a range of applications. I'm genuinely proud that our country can lay claim to so much ingenuity. Microbial metal recycling in Edinburgh, biosynthetic fuels from Manchester, lab-grown blood from Bristol, nitrogen-fixing cereals from Oxford, and nanocellulose-based materials right here in London. I want to end, though, on a broader point concerning emerging technologies such as engineering biology and others as well. Earlier, you heard me talk about risks and challenges, including the need for responsible innovation. But another challenge is about how we, as a society, talk about science and technology in general. Clearly, one of my aims this evening is to have a raising of awareness about engineering biology. But it strikes me that we're living through a period where public engagement around science is getting harder. And that's not just because of the unprecedented volumes of misinformation that circulate around us. We now live in a much less paternalistic society, which is surely a good thing. It is no longer enough for scientists to tell people what's good for them and expect them to toe the line. Instead, we know we need to have a proper, well-informed debate about these issues. Now, clearly, it would be possible for the promise of engineering biology to be thrown off course, to be derailed by public opposition. We need to listen to public concerns, really listen, and understand that if we don't respond to those concerns, people will be perfectly within their rights to not support or to actively block the engineering biology advances that we're trying to create. There is a lot of work to do here. I don't think we should ever be finished listening to the public. Essentially, the technologies being developed in engineering biology need to offer solutions to problems that people actually care about. Health, nutrition, climate, the environment, sustainability and global equity. I know that these are problems that billions of people do care about. I hope I've persuaded you that when it comes to these problems, engineering biology has solutions to offer. So thank you for listening. Do read our report, there it is, and thank you to the Royal Institution for asking me to speak in this 200th anniversary year for Discourses.